And of course, you see all your categories. You see receptors, enzymes, transcription factors, as well as conodes. If you actually click on a data point, you get a wealth of information about that particular experiment. Of course, you see the gene that's being interrogated. You see it was twofold upregulated. You see a p-value significance for that fold change, as well as a description of the biosample that was used, an experiment name, and here we're using a TLR1/2 ligand compared to a vehicle. So let's say, for example, you don't know what P3CKS4 is. We have links here to a bioactive small molecule, and what this does is gives you a link directly to PubChem. So you can find out exactly what P3CKS4 is if you're not already familiar with that molecule. And you can see from its pharmacology that it is a TLR2 and TLR1 receptor agonist. Additionally, what you can see from these data points, down here on the More Information button, is the experimental and data set information for that fold change. So you get the experimental name, which is the treatment versus vehicle here, a description of what that was actually done, the biosample, the species, and you can see what data set that experiment was a part of. Now, also, if you want to go directly and look at that data set to maybe see what other experiments were done, we now have a hyperlinked DOI that if you click it, takes you directly to the data set page, and all of the various experiments that were done in that data set. The one we were looking at happened to be experiment 2. All right. Let's go back to our page here. So so that was showing you evidence of the transcriptomic regulation of STAR-D10. And what I'd like to point out is here we see some evidence for nuclear receptor regulation by estrogen receptor. And here we see 2 blue data points, or in other words, downregulated. And if we click on our information, we happen to see that these are ER alpha knockdown experiments. We also see some experiments treated with 17 beta E2 and ER alpha ligand. And we can see that treated with ligand, these an upregulation of STAR-D10 in this specific cell line. So what we see is we definitely have some evidence of estrogen alpha regulation of STAR-D10. So now let's take a look at the Cystromics data set. So we recommend that when you switch searches like this, that you reset your filter. And so we're simply going to select single gene, but now we're going to filter on Cystromics only. Again, we put in a couple of keywords. We select our gene of interest here. Again, we're going to go default. We click submit. And what we see here now are a number of data points that show protein binding in the STAR-D10 promoter. So each one of these data points is a ChIP-seq data point for a particular protein. What I want you to notice are all these ER alpha data points in combination with various ligands and other proteins. So you can definitely see there's a lot of good evidence that ER alpha is binding the STAR-D10 promoter. Okay, so just looking at SPP alone, there is an abundant evidence that the estrogen receptor regulates STAR-D10. However, we could not find any mechanistic literature evidence, in PubMed for the estrogen receptor transcriptional regulation of STAR-D10. But what is known from the literature, however, is that pancreatic insulin content is actually regulated by the estrogen receptor. So if you take all of this information in total, And if you visualize star D10, the estrogen receptor and insulin as a 3 corners of a triangle, now the literature makes some connections, but SPP forms the third side of that triangle and allows the users to hypothesize a possible mechanistic basis for the ER insulin connection. And that hypothesis may that estrogen receptor is regulating insulin status in part due to its transcriptomic regulation of star D10. All right, so sometimes, these hypotheses can be difficult to generate if you only have a specific gene in mind. And so for this reason, we allow users to back their queries out, sort of zoom out, and encompass a broader range of cellular functions using Go terms. An example of hypothesis generation, again, of relevance to NIDDK, I'm going to pose here as an investigator seeking novel mechanistic evidence underlying the known link between HGF and brown adipose tissue formation. All right, so I'm going to go back to our live demonstration here, reset our filter, and we're going to enter in a Go term search, and here we're going to begin entering a couple of keywords to bring up brown fat cell differentiation. We'll select that. Now this serves as a proxy for a number of genes involved in brown cell fat cell differentiation. But know that on a run of a Go search, you need to specify the node class. And here we're going to select receptors. And this is to prevent potentially very large queries being submitted that might impact server performance. And so I've selected receptors and catalytic receptors. I'm going to click submit. And the default data display is the target display, meaning that these are all of the gene targets that were in the Go ontology term for brown adipose tissue formation that are also have evidence in SPP. Now, in order to narrow these down, what I can first do is I can select the category view. And what this does is now it breaks down those same data points into receptor families that may be involved. And so since I'm specifically interested in HGF receptors, I'm going to go back to our form, and I'm going to narrow down my search just to the HGF receptor family here and click submit. And so what this does is now I'm only looking at those brown fat cell data point genes, related genes that have something to do with HGF receptor, either down or upregulated in blue and red. And in the regulation report, I can see evidence for HGF receptor family regulation of PPARG gamma. Now, this is actually consistent with what is known in the literature, that PPARG gamma is regulated by HGF receptor family members. And so now this is actually kind of a positive control for my search, which gives me more confidence that the results I'm getting back from SPP are real, and that I can believe the other evidence that I see. So really what we really want to know is what are the novel HGF receptor family targets. And so what I want you to notice is that 2 different HGF receptor family ligands, TGF alpha and NRG1, both of these repress laminin 4 expression. Now, this is interesting in the context of evidence from the literature suggesting that laminin 4 is a negative regulator of brown fat tissue, of fat browning rather in mice. And so again, we have this complementary relationship between the literature and SPP, whereby the literature provides the outlines of the hypothesis and SPP provides evidence for the mechanistic basis of that hypothesis. Now, another way you can query SPP is by using your own gene set. So again, to illustrate this with an example of relevant to NIDDK, what I'm going to do is I'm going to pose as a researcher who's looking for novel receptor signaling paradigms in Crohn's disease. And as for my research, I'm going to take a set of genes that was identified in this paper to have prognostic value in Crohn's disease. So we'll go back to our search. We're going to reset it, and here we're going to select the option of gene list. We're going to click choose file so that we can select our Crohn's signature. Now, this is a comma-separated file of those genes implicated in Crohn's, their official gene symbols. So now I've uploaded my gene list, and I'm going to focus down here on catalytic receptors, hit submit. And what I see is a lot of evidence connecting various catalytic receptor signaling systems to transcriptional regulation of those genes implicated in Crohn's. So as a researcher in Crohn's, I already know that there's a documented relationship between HGF and Crohn's disease. So I'm going to explore that by narrowing down my search to just the HGF receptor family. And again, it's always nice to have positive controls, and here's one. So there's evidence that CXCR4 here is repressed by an inhibitor of the HGF receptor, right? And now this is consistent with what has been previously shown in the literature, and that is HGF regulates CXCR4, right? So now, what you also see, there are numerous other genes here that SPP provides evidence that are transcriptional targets of HGF. And one of these is the chemokine CXCL11, for which there is a large number of data points showing repression of CXCL11 in numerous independent experiments from actually the same inhibitor of HGF. Again, however, we were able to find no evidence for this regulatory relationship in the literature. So when we combine this with SPP regulatory evidence with the literature evidence that CXCL11 is consistently overexpressed in Crohn's, we've closed that triangle again on our hypothesis that HGF is connected to Crohn's, at least in part, through the induction of CXCL11.